Hello, how are you? I'm well. I'm excited about this chapter because it's a very interesting chapter to me. I like to read about viruses, and it's a very current and important topic now, right? Viruses have been changing our lives for so many decades and hundreds of years, but now we are more and more aware of how powerful they can be. It is the first time that we have a virus really changing our decision every morning. I hope it is changing how you think, and hope it makes you understand how important biology is, and microbiology mostly. So when we talk about viruses today, I want you to have in mind what's going on in the world and how if you have misconceptions about the pandemic that's going on, right? If you have any question about how the coronavirus multiplies in the body, because I think after reading and understanding this chapter, you should not have simple questions like how does it multiply, how does it enter the body, what happens during the multiplication, how that multiplication happens, and how does it exit a cell, what does it do next. So we're going to talk about all this today. Let's start with this. If you look at this scale, let me put my picture here so you can see what I'm doing. That's a coffee bean, right? If you zoom in, you start to see smaller and smaller and smaller particles. That's a protozoa, amoeba, right? That's a paramecium, another protozoa. A skin cell, an egg, a human egg. And things are just getting smaller and smaller. You see how we're now in 30 micrometers. Red blood cell is 8 micrometers. An yeast is 4 micrometers. Where is the virus? We're not close to the virus yet. Look at how big a bacterium is and a lysosome or a mitochondrion inside a cell are. We're still not seeing any virus. You see, that's the virus compared to the size of the bacterium. Look how different, how much smaller it is. Look how much we're going to have to zoom to finally see the viruses. Look at the size of this phage compared to the size of this bacterium. See how tiny they are? 30 nanometers, 45 nanometers. These are some animal cells viruses. And then you have the HIV is a little bigger. The influenza virus is bigger than the HIV. The measles is a big virus. So you see how tiny they are. So now I'm going to go back and you see, look, how minuscule they are. We cannot see a bacteria, and the bacteria is huge here compared to the virus. And they're still so powerful. They cannot duplicate by themselves. They don't have a cell, and they're still so powerful, right? So let's look at our learning objectives for today before we dig deep into this. So the first video, I'm going to talk about general features, viral structure, taxonomy, very little about how they grow in the lab. And then we're going to, in the second video, I'm going to talk about bacterial multiplication, meaning viral multiplication in bacteria. And then the third video, I'm going to talk about mammal multiplication. Okay? So be prepared. And then in the end, we're just going to mention the mechanism by which prions act. And prions are not viruses, right? So be prepared. This is a deep chapter. Involves understanding what is the DNA, how DNA duplicates, how RNA is made from DNA, and that type of information. So if you are not really good at that, make sure you go back and read it, okay? So remember that they are obligate intracellular parasites, so they cannot multiply and live outside of a host cell. They contain DNA or RNA, and here I'm gonna tell you, so we have either, we have both double-strand DNA and single-strand RNA. Viruses can have double-strand or single-strand DNA, double-strand or single-strand RNA, but it's either or. Very few exceptions have DNA and RNA. Viruses usually have DNA or RNA, okay? Single or double-strand. They contain a protein called capsid that I'm going to show you in a minute. They do not have ribosomes, they do not have organelles, and they do not generate any ATP. They do not have a metabolism. So comparing a bacterium to a virus, look how interesting this is. You have no multiplication in the virus, right? Where is it? No binary fission. I'm trying to get my pen here to work. Just got a new computer, so that's a good thing. So here, they can pass through biological filters, meaning it's really hard to remember filtration for sterilization that we talked about. You don't really get rid of viruses that way, okay? They do not multiply by binary fission. They can have either DNA or RNA, so they do not have both in the same cell. And you're like, how can they have RNA and still grow? It's crazy. We're going to talk about that. They don't have organelles. They're not sensitive to antimicrobials, and they are sensitive to a molecule from our immune system that we're going to learn later called interferon. Okay, so interferon is one way that our immune system uses to get rid of viruses. Host range. What is this? The spectrum of host cells a virus can infect, meaning some viruses are able to infect different types of cells, and some viruses have a very narrow host range where they only infect a specific type of cell or specific type of host, okay? There are viruses that only infect humans, for example, HIV. There are viruses that can infect us and that can infect, for example, birds, like the avian flu virus, okay? Just to give you an example of what I'm talking about here. When I say bacteriophages, remember the bacteriophage is the virus that infects bacteria, okay? Talking about this. So again, just to illustrate for you the sizes of viruses. So you can have very tiny viruses like these bacteriophages, and you can have much bigger viruses like the Ebola virus that is bigger than a chlamydia elementary body, which is a bacterium, right? I am not gonna have you memorize here the families of the virus, the types of the virus. We're just talking about viruses in general. We are gonna get deeper into names of viruses when we talk about the diseases, okay? So virion, what is that? It's one particle of virus. It's one unit of virus. So keep that in mind, okay? Capsid is the coat. It's made of capsomeres, which are the little units that make this coat. This is a capsid. And the capsid here is made of each one of these little structures, which are capsomeres. Okay, and the capsid is the whole thing. And where is the virus genetic material? Inside. Okay, inside the capsomere. Envelope. Envelope is made of phospholipids. It's a phospholipid bilayer. How come? they can be enveloped if, and actually the ones that are enveloped are the viruses that infect mammal cells or animal cells, okay? How can they be enveloped if they can even make the lipids? They steal it from the host cell. Their envelope is a piece of the plasma membrane of their host. And when I say host, you can think of humans, you can think about an animal, you can think about an insect, okay? Any animal can be infected by viruses. And then the third thing that we didn't talk about is the spikes. The spikes are protrusions from the envelope. So you see these little green things here? These are the spikes. The spikes are super important in vaccine making because the spikes, they're super external and they're protein made, protein based. So our immune system easily sees those spikes. Is that what they're using now to develop the coronavirus vaccine? Most of, or a big part of the vaccine production for the coronavirus is being made with a spike, okay? Because our immune system sees spikes of viruses really easily. Morphology, they can be helical, which is hollow cylindrical capsids, and the DNA or RNA is inside. They can be polyhedral, which is many-sided, a many-sided type of structure. They can be enveloped, like some of the ones that infect animals, and they can have very complicated structures and be called complex viruses. Again, you're not going to be required to know structures of a specific species. But for example, this is a bacteriophage. A bacteriophage is a complex virus, right? Look at that. It has a head, it has a body, and then it has in the body this base plate and tails, okay? So it is a very complex structure. The DNA goes inside the capsid, and all this needs to be assembled in order for the virus to be formed. So one of this is what we call one virion, okay? This is another virus, it's the orthopox virus. So this other virus has a totally different shape compared to the bacteriophage that infects bacteria. So when we talk about names or the nomenclature for viruses, remember that you don't have a kingdom and a domain, or again, a domain and a kingdom or a phylum where viruses are included. So the first class that you have for viruses is order, and then you have family and genus. And the names of the orders end with else, families end with viridi, and the name of the genus end with virus. You do not have species, okay? So you usually have one genus, and that's the last classification. The species, what I'm trying to say, you don't have a name for species, okay? The name of the virus is usually the genus of the virus. A virus species is a group of viruses that share the same genetic information and same ecological niche, meaning which host they tend to infect. So there are two ways of growing viruses in the lab, depending on if it's a bacterial virus or an animal virus. The bacterial viruses or the bacteriophages can grow on a plate. But what do you need to put on this plate? You need to put bacteria. If you just spread viruses on these plates, it's never going to grow. But when you put bacteria and then you add the viruses with the bacteria, you see that they form these plaques. So the plaques are formed wherever the bacteria die. This is death. That's why there are these holes in the sheath of bacterial growth, right? And the lawn that is formed. So wherever there is death, you know that a virus, that there was a bacterium or a group of bacteria infected by viruses, and that cell lysed, and all the virus were spread in the environment in that specific area, okay? So this is a very efficient way to grow bacteriophages, and it's not that hard when you compare with animal viruses. Those have to be grown in living animals, or if you want to make it a little simpler, in embryonated eggs. So you inject the virus into the egg, and they grow in different parts. You can inject them in different parts of the egg, okay? And because the egg is made of cells, they infect the cells, they use the cell machinery, and that's how they grow. And then they can be extracted. In cell cultures is another way to grow virus. So you know how you can have tissue culture where you isolate animal cells and you put in a very enriched growth media and you can grow them there. And cells that are transformed, I'm trying to make this bigger for you to see, hold on. Cells that are transformed by viruses are going to look like this. You see, they're going to be able to multiply and not die, because the normal cells in vitro, at some point they're gonna die, but the transformed cells don't die. Why? Because remember how viruses have this ability to multiply infinitely, so they just make that cell immortal, and then that cell multiplies. Actually, adding viruses to a cell is a way to establish a cell lineage, which is a lineage of cell that is never gonna die, okay? You can freeze, you can thaw, you can grow, they keep growing, okay? All right, what else I wanna tell you about this? This is cool. Viral identification. So, I'm gonna put my, so how do you identify if there are viruses growing inside a cell? There are cytopathic effects. The cell looks sick. One thing that happens to the cell is this, where you have many cells fusing together because they're infected by virus, and the nuclei get all like together because all those cells fuse. So you call a multinucleated cell. This is a very typical effect of viral making a cell sick, okay? There are serological tests that can be done with Western blotting, which is a way to detect proteins, and also ways to measure nucleic acid, which are super efficient. So when you think about nucleic acid detection, you can use RFLPs, which is one method to detect it, but the method that we usually talk about here is the PCR, which is polymerase chain reaction, okay? So polymerase, remember, is the name of the enzyme that duplicates the DNA. So what's gonna happen here is that your DNA, so this is the DNA, right? You start with one copy. That one copy is going to be multiplied in vitro because you add everything you need. If you look at the list here, later you can check, and it says everything you add to this little recipe to make the DNA duplicate. And when you put this little tube in this machine that is called thermocycler, this machine is going to decrease the temperature to the optimal temperature for the enzymes that act in DNA duplication. Remember, DNA gyrase, DNA ligase, DNA polymerase, they are gonna act at their specific order because you change the temperature to the temperature that you want that enzyme to act. So if first you want the gyrase to despiralize the DNA, you use the temperature for that one. And then you increase for the temperature of the polymerase, and then that one is going to be the one is going to act. So with the temperature game, you can synchronize that synthesis of one DNA from a template. Okay, so from one template, you make 2 copies. From each one of these 2 copies, you make 4 copies. And then you keep multiplying. Why are you amplifying, multiplying the DNA? Don't you just want to know if this is a virus or not? So what you're doing here is you're actually amplifying a region of that virus that is known, right? So if I know I'm amplifying a specific region of a DNA virus, I amplify that. And if that really amplifies, that means there was a template there. If there is no amplification in the end, it's because there was no virus there, because I used a primer for, which is a specific piece of DNA that is just going to bind to that viral DNA. It's not going to bind to my, regular DNA from my cell. That's not going to happen. So I know that I'm specifically amplifying that piece of DNA. That's how specific this method is, and that's why it's so important in, for example, detecting if somebody has the virus. We're not talking about antibody here. We're talking about if you have the virus against that is causing a certain disease, okay? You're determining viral load here. Because also by this method, you can then do a calculation and tell if how many copies you had in the beginning, if in the end you had X number of copies, right? You can make a calculation to know how many you started with. When we think about how viruses multiply, we always think about a one-step curve. So this curve, which says number of viruses here in time in days here, what is this telling you? Well, there is an eclipse period. What is this U-shaped part of the curve? Why do you get to 0 when you started with a specific number of virions? How does it drop to 0? Think about the virus we're going to learn in a minute, that the virus need to disassemble in order to multiply all the parts and reassemble. So the disassembling in that moment, if you take a picture of that moment of that whole cell with viruses inside, all the viral particles are disassembled. So how many complete particles do you have? None. Okay, that's why it goes down to 0 here. And then the virus all together, all the virions start to get assembled again. So you start to multiply the DNA, the capsomere proteins, if it's a bacteriophage, all the other parts of the bacteriophage, and all that gets assembled, which is the maturation step. And then you have that peak. So they all multiply at the same time. That's why you have an acute infection. Okay, and then these viruses leave the cell, and it gets to a point that, you're talking about infection. So your immune system is going to learn how to detect and kill those infected cells. And then you have the drop in the number of viruses during that infection. Okay. Okay, so that's it. We're going to start to talk about multiplication of bacteriophages inside bacteria in the next video. I will talk to you soon. Bye.